Okay, good afternoon, and welcome to the second lecture in the 2018 fMRI summer course. My name is Sean Marrett, and I'm a staff scientist who works for Peter Benettini in the core facility with sort of broad responsibilities in neuroscience and stimulus devices and things like that. I've given a version of this lecture for the last couple of years, and the basic goal is to give you an overview of the functional MRI facility, the resources that are there for those of you who may be new to the NIH, and some history of the MR research that's taken place there, as well as sort of what I call a little bit of a postcard tour of some of the work that is ongoing, what some of the different PIs are doing. And I doubt that you'll take advantage of this, but I'll just say if you have any questions, don't hesitate to ask. And part of the motivation here is just for you to realize we're actually it's really a vast research enterprise that we have. It's probably one of the largest MR research facilities in the world, and we have more than 300 people who pay attention to, or at least receive our emails. There's 80 protocols. There is a vast array of research that's going on there, but also support work, and that may be helpful to you if you have some basically burning research questions. Maybe somebody else has some ideas, and you can always come and talk to us and ask us if somebody has done something, whether you're interested in delivering tastants or delivering painful stimulation, different ideas about visual stimulus, different ideas about experimental paradigms. We try to basically facilitate as much communication in the community as possible. So just quickly, how many people here have actually already done an MRI experiment? Are you all sort of experienced fMRI folks? Yes? Okay, most of you are, say 80%. And how many of you have actually been in the MRI scanner? Everybody? No? Okay, interesting. All right, I suggest it's a good thing to do if you have a chance, just to get an idea of what you're putting your subjects through. So, basic outline, I'm just going to give you an overview of the neuroimaging resources as well as the MR resources. And that is not only neuroimaging, but it's also things like the AFNI folks, MEG. There are some people, if you're ever interested in building specialized devices, section on instrumentation that you might be interested in, and a couple of the other toys that we have around. I'm going to give you some history of brain MRI at the NIH and some of the examples of some of the, I think, really foundational methods developed here, and an overview of the research, and then this postcard presentation of work by selected fMRI users, PIs. And just a couple of short points. It may not be obvious to you, but this really is one of the largest concentrations of MR resources just about anywhere in the world. And it's a little bit daunting when you think about how much MR research has been done here. I mean, you pass people in the halls who have developed methods for arterial spin labeling, for the initial methods for analyzing MRI, for arterial spin labeling, and there are resources to just about anything here, from developing pulse sequences to new instrumentation, new gradient coils, new RF coils, you name it. There's someone who's probably trying to do it. So first off, just to sort of point out that we're not alone. We've got all sorts of other neuroimaging resources at the NIH. You have the A team here, the AFNI group, the Scientific Statistical Computing Core. Those are people who can often help you analyze your data. And I'm not going to talk about animal imaging, but there is a very extensive animal imaging facility here, both for small rodents, mice, and rats. And they're actually, I'll point out when I put a floor plan of our facility later on where they are. There is sorry, let me just get used to this. People who can help you build, that may be a little bit small here, but basically there's a group that can help you build all sorts of different devices, and we rely on them for building mirrors, gustometers, all the different devices that we use for providing instrumentation to record behavior in the magnet, and there are just an incredible team of people who both can help you with machining, 3D design, CAD, electronics, you name it. And that's called the section on instrumentation, led by George Dold. There are some new groups that you may be aware of. There's a data science and sharing team led by Adam Thomas that is really doing great guns, making it possible and easier for people to share their data, as well as standardize the way that data is accessed, making it available to people outside the halls of the NIH, and also bringing in large data sets so that you can, things like the brain bank data from the UK has been made available on the BioWolf. In addition, there's a new machine learning team led by Francisco Pereira that has just really come online and is just doing a lot of work to make those kinds of methods available for doing using deep learning and other methods, Searchlight and so on, to analyze fMRI data, but also other behavioral data as well, and other general neuroscience data. And finally, in case you don't know, we also are home to one of the largest scientific computing clusters around, and it's an amazing resource. And if you don't have an account on that BioWolf, I suggest you go get yourself one. It really is can make a huge difference to your productivity. Now, in addition to those sort of non-imaging, we have a sister facility, which is the MAG, which is led by Alison Nugent, pictured here, and they have a 275-channel state-of-the-art MEG right next to 3TA, which is really an amazing resource. And there is a MR spectroscopy group led by Jun Shen, pictured here, in case you decide that you're interested in looking at what's going on chemically in the brain besides just simple bold measurements around anatomy. And I've got a picture here of Pete Malfeas. We also do simultaneous EEG. We have a system from EGI as well as a system from Brain Products. The pictured here is a 256-channel EGI system, which we recently upgraded. And in addition to that, the system also has an amplifier that can allow it to be used for doing neuromodulation. So why do I love working at the NIH in fMRI? I think there's a couple of reasons. It has this amazing history of developing all these different techniques, looking at contrast mechanisms, people who basically have the time to just look at all these questions in amazing detail, from the basis of the bold signal to what is it that actually creates contrast in T1 images. You have this amazing range of technical expertise for coil design, pulse sequence development, novel experimental paradigms, development of things like decoding, and ICA for multi-echo. And then there's just this amazing array of clinical and basic neuroscience questions that are just constantly being investigated. And I'll just reiterate here that it really is hard to know everything that's going on in our facility, but you should not feel shy about asking us if you need help, if you have any question. Often we won't know the answer, but we will know who you can talk to. I was asked also just to mention the fact that there is a lot of work going on in neuromodulation, and it's worth pointing out that some of these people here were at the beginning of the development of transcranial magnetic stimulation in the 1990s, and people like Andre Pascal Luoni did his training here, research training here. This is Leonardo Cohen, and Eric Wasserman. And just recently, the NIMH has had Holly Lisenby join us as the Director of Translational Research in the extramural program, but she's also running a new non-invasive neuromodulation unit, which is we're just in the process of installing a new simultaneous TMS fMRI system on one of our scanners. In addition, there's all sorts of people who are doing neuromodulation using TMS both for treatment as well as for looking at cognitive processing. So that includes people like Carlos Zarate and Chris Baker and Leslie Ungerleider and others. So I just put here a list of some of the scientific and technical innovations that have were developed at the NIH, and this includes just the first look at looking at the natural history of neuroimmune disorders, multiple sclerosis, that started in 1989. The first studies using EPI for functional MRI were done. Some of them were done here as early as 1992. Diffusion-weighted imaging and diffusion tensor imaging. One can argue about it, but I think you could you can make a really good argument that it was actually developed here in the early 1990s by Peter Bassert and Danny Liebehahn. The longitudinal studies of normal and clinical brain development, especially in pediatric populations, that's something that started here almost 30 years ago by Judy Rappaport and Jay Geed, and some of those studies are ongoing. In fact, some of the kids who were studied back then in the 1990s are being taken back and studied again as adults by Armin Resnahan. We've had impact on gradient coil design, on the basically the use of parallel receivers. This is a method by which, back in the old days, not that long ago, you're all used to using 32-channel coils now, but there was a time when the coils just had a single channel, and methods for allowing you to improve the SNR were developed here and implemented here practically by people like Jeff Dunn and others. We've played a role in learning how to practically use multi-echo bold for denoising using ICA, and a lot of the original work on fMRI decoding was done here by Jim Haxby, and informational mapping by Nico Kriegersport, and then we've done a lot of work on ultra-high field, anatomical imaging, and most recently using very high resolution laminar fMRI using cerebral blood volume methods such as VASO at ultra-high field. You're going to hear about all these things later on in the course. So the facility was established in 1987. The first studies were done in animals. The first functional studies were done in animals in the late 80s. There was a Ford Tesla that was actually built here. It was one of the first 4Ts. I think it required an actual act of Congress. That was done by Bob Balaban, who a lot of that work was actually, even though they built a special head coil, it was done by the Heart, Lung, and Blood Institute. So that's, again, an instance, I think, of where this sort of broad, long-term thinking and consistent resource availability has made it possible for a lot of cross-institute and cross-scientific fertilization to take place. Yeah, so this is just a list. This is when things started up. There was Ted Becker was essentially the head of intramural research, who hired a very famous physicist by the name of David Holt from the UK, who hired Bob Turner, who is a really fascinating character in functional MRI research, who got his major kick to his career here. And Bob Turner collaborated with someone named Danny Liebehahn, and they worked very hard on establishing diffusion-weighted imaging on this 1.5 Tesla scanner. And then Henry McFarland, a neurologist in NINDS, began this long-running natural history protocol, which is still recruiting patients almost 30 years later. Henry is... Harold has retired, or it's Henry actually, not Harold. But Danny Reich is still using that protocol, and they're keeping the scanners busy with the subjects in that. So this is a newspaper clipping announcing the opening of this new NMR center. I won't mention names to identified the guilty, but there are people here who are still active researchers. This was our first 1.5T scanner, unshielded. You wouldn't want to use a color monitor outside, but they managed to get this facility built in 2 years from when they first idea to when the system was installed, and they scanned their first human, which is, I think, a record in terms of how fast things could get done then, especially if you're familiar with how long construction projects take here. So just a couple of the personalities here. This is, I mentioned Bob Turner. He was here for several years and was instrumental in getting some of the first high-field EPI images from the visual cortex. This is this incredible 4T system that was built here, and he designed a special gradient coil so that you could actually get the kinds of fast gradients you needed to do single-shot EPI. And the person here putting this unsuspecting subject into this massive magnet is Peter Jezzard, who runs now the physics group at Oxford, who was here as, I guess, as a postdoc at the time. And I will just make a small advertisement that if you really want to get a sense and read a highly entertaining paper, this paper by Bob Turner that was published in 2012 in Neuroimaging, really just gives, it is absolutely fun and fantastic to read. It's completely hilarious. He is completely unself-conscious in terms of his style of writing, and he just gives amazing anecdotes, so it's a great paper. I'll just mention one other thing that was happening back then that I think is worth dwelling on. This is Peter Bazzer, who is a principal investigator now in NICHD, and he was instrumental in developing what's called diffusion tensor imaging, which is a method for diffusion imaging. You're looking at the diffusion of water molecules in different tissue types. So you can use it to identify, for instance, if someone had this may be an example of someone who's had an infarct of some kind, and you see a hyperintensity in the diffusion image. He had the idea that you could actually start looking at different directions of diffusion in the brain, and this eventually led to the ability to look at white matter connectivity, which we all take for granted now. But this is a method that was developed here, and if you go back and look at the papers that he wrote in the 1990s, they've been cited over 2,000 times now. And I'm just going to mention two other people who have been, I think, responsible for a lot of the reasons that we have such a successful MR program. Alan Kretsky is about to step down as scientific director, but he is also being the director of our what's called the In Vivo NMR Center, which is basically the facility that houses the functional MRI facility. And he's been here since 1999, but I think he actually did his postdoc with Bob Balaban, who is the scientific director of NHLBI. He's famous for developing all sorts of methods in MRI, especially magnetization transfer, which is a method for looking at chemical composition in living tissue. Both of these folks have been given the gold medal by the International Society for Magnetic Resonance Medicine, independently of their organizational and bureaucratic achievements in terms of supporting magnetic resonance imaging research here. But if you see them in the hall, you'll know who they are. All right, I think I'm going to skip quickly over this. But the main point here is Alan Kretsky there's all this initial work really ramping things up in the 1990s. Then Leslie Ungerleider got very interested in using fMRI for looking at normal cognition and created a group that initially was using positron emission tomography, but then switched over to fMRI. Alan Kretsky was hired to run the NMR facility, and the key thing about that is that he basically pushed us into purchasing a 3 Tesla scanner rather than multiple 1.5 Ts. And I think that was at the time, 1.5 T was kind of a standard system for collecting MRI data. And I think it was a very fortunate decision that he decided to go with a 3T. Peter Bannattini, my boss, was hired to run this new facility, and we took delivery. And essentially in 2000 is when we began routine scanning on our first 3 Tesla scanner. With a lot of people were hired in the ensuing years, and there's a big programs built up, especially in mental health, that depended on functional MRI. And we bought our second scanner, and we also did a lot of technical development on parallel imaging in collaboration between Jeff Dunn and Peter Bannattini. And what was important about that was showing the utility of doing parallel imaging, which I'll show some images of a little bit at 3T. Yeah, so we increased the number of scanners, and we have now gone. Our first 7 Tesla was installed in 2002, but it was very much an experimental system. And in 2011, we purchased the first self-shielded system from a combination of a couple of vendors. We basically assembled it ourselves, and that became the basis of the Siemens 7T product, which has now led to something which is now FDA cleared. But that has become a very popular system indeed. We also got an 11.7 Tesla scanner, which sadly quenched and has been spending time in England and Italy, but we're hoping to get it back at the end of this year. We hope to have it back and running again. So just to give you an idea then, so we've gone from 1.5 T now to 6 3T scanners just within our facility, as well as the NIDA. We've now got one 7T and another one about to be installed, and hopefully by the end of this year we'll have an 11.7. So there's just been an amazing growth in our facility. As I mentioned, we have over 300 users. We're doing about 7,000 exams or subjects per year. And of course, the number of papers that are published that just mention MRI and come from Bethesda is astonishingly high, and we have now published more than 1,200 papers that use the facilities of the functional MRI facility. We've got an H-index of over 150 and 92,000 citations. So. If you just wanted to keep up with all the different papers that were being published using the facility, I guess you would have to read about 2 to 3 papers a week. All right, so most of you may be aware of this, but this is a floor plan of the functional MRI facility and the NMR facility. So the scanners, the things that are in sort of brown here belong to or have the name of the NIMH on them. So this is 3TA, 3TB, 3TC, R7T, and 3TD. And then the NMRF 3T, which is in the process of becoming a 7T for those of you who spend time downstairs. You'll notice a construction project there. We're expecting that'll be operational and turned over to us by the end of this year. There's a Prisma 3T over here, which is a high-performance scanner from Siemens, which has the NIMH and NINDS together have about a 50% stake in that scanner. And over here is this animal imaging facility, the mouse imaging facility that I was referring to earlier. So there's multiple high-field scanners there for doing in vivo imaging, as well as ultrasound and biofluorescence and CT scanners for small animals. So if you are interested in doing small animal imaging, we're extremely well equipped for that. That's sort of a sister facility. This is the 11.7 bay, which is essentially a storage unit right now, but we're hoping that it is going to be up and at field by the end of 2018, maybe the beginning of 2019. We'll see. So we've got basically three different models of scanner. We have 3 GE scanners, all 3T. They're all essentially now the same model. We've upgraded all of them to the MR750, which is sort of the modern platform for GE. Almost all the studies that are being done here now use the GE 32-channel head coil, and they're absolutely stable. They're relatively state-of-the-art, and we are hoping to do a special project on this, which is going to make this more state-of-the-art with a dedicated head gradient, but I'll let one of my colleagues talk about that at a later date. 3TD is a wide-body Siemens Skyra, not quite as high performance, but very, very flexible. And then, of course, there's the 7T, which is very popular for doing high-resolution, high-field studies. And I guess one would consider it a success that since we have purchased one, we're going to be one of the few facilities that is going to have a second 7T available for doing studies in normals and patients by the end of this year. Okay, so that's sort of the conclusion of the facilities. If you've got any questions, I'm going to just start doing a bit of a postcard tour of some of the researchers here. But if you have any questions, this would be a good time to ask. Otherwise, I'll just continue on. Nope. All right. Okay. Oh, yeah, sorry. Yeah, the Prisma. Pardon me. Yeah, that's a slightly higher performance system. It's one nice thing it has is a 64-channel coil, which is a higher coil count than any of the other scanners we've got, which provides really good coverage down the neck and is slightly larger than the standard Siemens 32-channel coil, which makes it convenient for doing simultaneous EEG and MRI. And again, as I say, we have between NIMH and NINDS, we basically have a 50% stake in that scanner. All right. So I think I mentioned earlier that we started doing longitudinal pediatric studies of brain development in kids back in the early 1990s. And although Judy Rappaport is no longer actively collecting data, and Jay Geed, who began those studies back in the 1990s, has now gone on to sunnier climates in Southern California. That work is being continued by Armin Rasnahan, who's basically doing a lot of really fascinating longitudinal studies of brain development, and he specializes in linking genetics and environment to brain development. They actually have just a recent paper that just showed up in Science looking at how different brain structures vary with brain size, which I find fascinating. I haven't read it fully. He is very, very careful about his methodology, and he is the one who has been bringing these subjects back after 20 years away, 15 years away, for what they call the take-back study, so they can actually compare the data collected in the 1990s to data collected now. So you see the whole trajectory from child to adult. He's got a whole other world of looking at sex-linked genes that I'm less familiar with. Another person who came out of that sort of whole school of looking at longitudinal brain development is Phil Shaw. He did a postdoc here, or a staff fellowship, I guess he was a staff clinician in NIMH, looking and did some papers looking at intellectual development and brain structure. But more recently, he's moved over to Human Genome Research Institute, where he is focused very much on children with ADHD, looking at the development of the brain of children with ADHD and looking at individual differences and seeing how the trajectory changes between kids who are at risk and who develop ADHD. It's sort of a unique collaboration. It's very, very focused on the influence, the interaction between genes and brain development, as well as environment. And in fact, if I'm not mistaken, I believe he's going to be starting a neurofeedback study soon enough to potentially see how neurofeedback can be used to potentially help kids who have ADHD. Karen Berman is another person who is very interested in the interaction between genes and brain development. She has been here for a long time. Her specialty is developmental neuropsychiatric disorders. She has been looking at the genetics of social cognition. They look at these patients, these young kids who often suffer from something called Williams syndrome, who they are delightful children. They have some intellectual disability, but they are hypersocial. They're just incredibly friendly. And they've done just first-class work looking at that. She also has been taken over the efforts at looking at developmental neuropsychiatric disorders, especially schizophrenia. And Karen's also got a program looking at a collaboration with Peter Schmidt, looking at the influence of puberty on brain development as well. Jeff Dunn is an NINDS investigator who is extremely well known and respected for the development of different imaging methods, both at ultra-high field, especially at ultra-high field, I should say, and has looked at a lot of the contrast mechanisms that make up why myelin changes the signal in T1 weighted images. He single-handedly sparked a huge amount of interest in looking at phase contrast in ultra-high field, and he has a group which has been working hard on developing new coil technology as well. They developed some of the first some of the designs they use for parallel receivers for the brain, basically turned into commercial products. They've got a whole new design, and he's the lead on the 11.7 project. And they've got a whole new design for doing improving the performance of high field receivers. And in addition to that, he's been very interested in the basis of the spontaneous brain activity in terms of the fMRI signal, and conducted, I think, one of the most rigorous studies of sleep, where he had one of his staff scientists basically have people spend 8 hours in the magnet having EEG measured at the same time as they're measuring fMRI for 8 hours. Really just a technical tour de force as well as a methodological tour de force. This is some of the work they did back in the day, showing the development of these parallel receivers. You can see the coils opened up, and you can see the coil elements, and showing the improvement in TSNR when you use the parallel receiver technology. This may not seem like much now, but 15 years ago, this was a big deal. Mark Hallett is again he's an extremely well-known neurologist working in movement disorders, and has been a user of fMRI in the facility since its inception. Very active in using neurofeedback and doing simultaneous EEG. And I should also mention that his people who trained with him were also he was one of the first people to really advance the whole area of neuromodulation, especially TMS, back in the early 1990s and early 1990s with people like Pascal Lyon and Leonardo Cohen, as well as Eric Wasserman. Another veteran is Leslie Ungerleider, who's been here, a very early adapter. I think arguably she's one of the people responsible for advocating for a dedicated functional MRI facility. What's unique about her, she has an interest in the architecture of the visual and attention systems, but I think she is unique in having a combined non-human primate and human imaging study, which is really something that is not very common. It's sort of it's quite demanding. So she uses both our facility as well as the neurophysiology imaging facility over in Building 49. She's extremely well known and well respected, and has just been doing fMRI for a good long time, and she's the chief of the laboratory Brain and Cognition. So one of her colleagues is Alex Martin, who has a section there. Alex is more focused on the neural foundations of memory and social cognition. He is a specialist in looking at how objects and categories are represented in the brain. And recently he's actually been working, some people working with him have been using neurofeedback to look at what happens with autistics and seeing if you can use completely new types of paradigms for doing neurofeedback, which shows some promise. And there's a really nice paper by Michel Rameau in 2017 that's worth a peek. I'm just going to say one other thing about Alex. This is some work that he did with Mike Beauchamp, and I always think of this as kind of the canonical argument for why you want to do high resolution. And what they demonstrated was that this was the application of that parallel receiver system that I showed a figure of from Jeff Dunn. And what they showed here was that these areas in the superior temporal sulcus that people had thought were multimodal were actually, when you examine them at high resolution, and remember this is at 3 Tesla, actually there were separate patches that some were visual, some were auditory, and only a very small number were multisensory. So if you looked at these patches with a voxel size of 3 millimeters, you just saw sort of one multisensory patch. But when you went to a 1.5 cubic millimeters voxel size, you were actually able to distinguish these separate patches in STS in superior temporal sulcus. And I always think of that as being really a classic example of the advantages of doing high resolution. These were not easy studies to do, and of course this was a home-built coil, but it was really, I think, a very elegant piece of work. What can I say about this guy? He's my boss. He is a phenomenon in many different ways. In addition to being editor-in-chief of Neuroimage for 7 years, he also ran our facility and runs a section. His research interests, I guess you heard him talk on Friday, is basically maximizing the information extracted from fMRI time series. He's worked on using multi-echo EPI for improving fMRI and clustering and understanding the mechanisms. He did pioneering work on information mapping and decoding of fMRI, and most recently, postdocs working with him have been looking at layer-specific imaging using vaso techniques, CBV-based methods for improving the layer specificity. I think this is some work he did with Javier Gonzalez Castillo, showing that when you averaged over massive numbers of runs on a single subject, you could actually show that there was activation just about everywhere in the brain with a simple visual stimulus. So that has been well-cited work. So he's just this very creative and, as you say, extremely energetic investigator. Yeah, I won't rest on that. That's mostly talking about his work looking at the physiological underpinnings of the resting state signal. So there's groups also, there's another set of researchers in NIMH who are looking at, I guess, mostly pediatric adolescents. This is Danny Pine, who runs the emotional development branch. He's very interested in the development of anxiety and looking at individual differences and what can influence treatment outcomes with kids with anxiety. He works closely with Ellen Liebenluft, who looks at bipolar disorders, but also has been looking at irritability in basically the levels of different, how fMRI can be used to distinguish between different patient groups who suffer from different types of irritability. And they have someone who's just joined their group is Argyris Stringaris, who I didn't make a slide for, but he is looking at the development of anxiety and especially looking at reward processing and using sophisticated computational techniques for doing that. Other users of our facility include Catherine Bushnell, who runs the National Center for Integrative Health scientific program on campus, and she is a very well-known pain researcher, and she has recruited Lauren Atlas, who just did her first BSD, so she's been here for 4 years. These folks are looking very creative. They're using these thermal pain stimulators in the magnet, these slightly terrifying devices that you can use to not burn people, but give them a little bit of pain sensation, doing eye tracking, and also looking at rewards. Now they're using tastants, using a gustometer to deliver different juices as rewards for changing expectation and pain perception. All right. Okay, and then Carlos Zarate, who runs the experimental therapeutics branch in NIMH, who's a very active user of simultaneous EEG fMRI, looking at fast-acting antidepressants and different types of neuromodulation, and has done some functional MRS as well in the magnet. And he has actually they are at one of the groups that's doing patient studies in our actively shielded 7T. These are patients who are given drugs in the magnet. It's technically demanding work to be doing that in the 7T environment. And I guess one could say that he's very interested in sort of neuromodulation, both in terms of fast-acting drugs as well as TMS as potential treatment for major depressive disorders. All right, so this is just a quick tour through the 7T and just a demonstration of how robust the images are, the functional contrast that you can get from the images. And this is some work from Laurentius Huber, you'll see, who's demonstrating that you can get laminar differences in functional MRI using this vaso technique, which is very, very hard to do with bold, showing this really nice distinction where you don't get this sort of change in peel vessels, which can sometimes confound the images so that you see things that maybe are in the sulcus rather than in the gray matter. So this is work that's been done by Laurentius under the direction of Peter Banatini. And other users of the 7T include Chris Baker, who is someone in the Laboratory of Brain and Cognition, who is looking at mostly face and scene representation and basically interested in learning and plasticity and how these two things interact both bottom up and top down. And the last person I will mention on the 7T is Danny Reich, who's a neurologist in neuroimmunology. And I think I mentioned that there was someone who was still using a protocol that was written in 1989. So Danny is the steward of this natural history study of people with multiple sclerosis, and has really done, he's been very interested in what the pathophysiology of MS lesions are, and he's been using the 7T to examine what he calls the central vein hypothesis, the idea that you basically, lesions are actually formed at the periphery of central veins in the brain, in white matter. And they've also been looking at whether or not some of the symptoms that one suffers from when you have MS actually may be coming from lesions which are not easily detected at 3T, but more easily detected at 7T in the gray matter. So some of the behavioral issues that may arise may only be visible when you look at it with high resolution and very high sensitivity. Okay. Last person, I guess, not quite last person, but Christian Grion, who is looking at the neurological basis of anxiety and fear, and they've recently started using the 7T for a lot of their studies, showing that the, I think it stands for the bed nucleus, the stria terminalis. Basically, they're showing changes there and connectivity there that you cannot see at 3 Tesla. All right, and I'll just, this is Eli Merriam, who is another active user of the 7T, very interested in very high resolution studies, the striate cortex mostly, and V1 early visual areas, and has recently joined the Laboratory of Brain and Cognition. So that's it. This is just an advertisement. This is what the nice new spanking 7T Terra from Siemens will look like when it arrives in September, and they now have FDA clearance for this. Doesn't make a huge difference for us, but I think it is kind of neat that we're going to have 2 7Ts. And I think that's a wrap. If you have any questions, feel free. Otherwise, you should all, anytime you just have a question, as I say, there is this amazing array of resources here, and it's hard to keep it organized in my own head, as you can see, but I'm very happy to share any information or anything I can do to help you get your studies done. All right, thank you very much.